Today I'll tell you about what I'll call a case study in reproducible data science. My idea is that this kind of process can be applied to many different fields of science. So if you come from neuroscience, this will be interesting because I'm going to be talking about brains. But if you don't, if you come from some other field and you're interested in thinking about how we might make reproducible data science in our science in whatever field, you can think of how this would apply to many other kinds of data. And I'll talk about a process that has, in my opinion, 3 different parts. The first is really about measuring, modeling, and explaining, and has to do with the substantive knowledge of the particular system that you're looking at, whether it be stars, weather patterns, or small things, really small things. And then the second has to do really more with evaluating what you've learned about the system. So if you create models of whatever data, you want to know how well are those models explaining the data that you've measured? How well are they accounting for the phenomena? And then the third has to do really more with reproducibility and sustainability. And is maybe more of an engineering. There are aspects of this that are social engineering, but some aspects that are software engineering. How do we make this kind of effort something that other people can build upon once we've got results? How can others just take those results and continue to do the research? And I'll talk specifically about the problem of assessing network connectivity that we can measure in the living human brain. So let me talk about, show you what that might look like. So here's an estimate of the connections in a single human brain measured while this person was alive, still is alive. And we refer to the sum total of all the connections in the human brain. We refer to that as the human connectome, an analogy to a genome or something of that sort. And what we're looking at here is these lines, these tubes that you're looking at are estimates of the trajectories of big bundles of fibers of the axons that are the projections of neurons through the white matter of the cortex. So the cortex is made out of gray matter, which covers the outside. That's where a lot of the cell bodies of the neurons that make up our brain lie. And then there's a big mass of what we call white matter in between those gray matter. And inside that white matter is where the trajectories of the connections between the different parts of the brain go. And so these colorful bundles of spaghetti that you're looking at are the branches that form the network of connections, that form the network that is our brain. And we want to measure those using magnetic resonance imaging. In fact, magnetic resonance imaging is probably the only way that we can measure that in a living human right now. And MRI can be used to measure many different kinds of things. You can measure some kind of estimate of neural activity using what's called functional MRI. You can measure the anatomy of the brain, the structure that I showed you on the outside there, using what we call maybe structural MRI. And there are many other things you can measure with MRI. Today's talk, I'll be focusing on measurements of connectivity using what we call diffusion MRI. So I'll go into a little bit more detail about how we make this measurement in just a minute. But first, I'll try to impress upon you that we think that the white matter matters, that it's actually important to make these measurements. And it's actually been accepted for a long time that there are many different kinds of situations in which people might have different kinds of disorders that come from disconnectivity. So things go wrong somehow in these connections in the white matter, and that might lead to, for example, an inability to read or inability to identify objects. And this was appreciated way back in the 19th century by these neurologists whose pictures are up here. And the reason for that is that these are not just passive cables that transmit information. The white matter actually changes with development. It adapts with learning. It affects our cognition, our behavior, and various diseases that affect the white matter. And then if you measure in different individuals, you might be able to explain a substantial amount of variance in how they behave by looking at differences in their white matter. So it's pretty important to measure that. And the main principle on which diffusion MRI relies is the fact that the tissue structure itself restricts the diffusion of water. So if we look at a cell, say something spherical like this diagram of a cell, we look at the water molecules in there. These water molecules can freely diffuse inside of this cell, but they're actually restricted in their diffusion by the boundaries, the membranes of the cell. And we rely on measurements of this microscopic diffusion in diffusion MRI. When water is present inside of something that's spherical like this, it might be diffusing in the same rate in all different directions. And we refer to that as isotropic diffusion, same in all directions. But if we're looking at an axon, one of these connections between the different neurons in the brain, which may be on the order of maybe 1 to 10 microns in size, we look at a water molecule that's inside one of these axons, it is actually restricted to diffuse more along the length of this axon, or along the length of the connection, than across the boundaries. And so diffusion in this case is anisotropic. And if there are more of these bundles or more of these axons that are together in a bundle, and if this bundle has, say, some kind of isolating material that we call myelin that covers these axons, then water that's actually outside of the cells will also be restricted in the diffusion. So if we measure along the length of one of these bundles, we'll find anisotropic diffusion. And that's what we measure in diffusion MRI. For example, if we look at one connection, you can see here, this is a brain in a postmortem dissection. What's happened here is that the part of the brain that was covering these bundles here was actually removed in the dissection, so that it uncovers these bundles, these large bundles of axons. And you can see that they fasciculate into these fairly large tubes. These tubes are not the individual axons. These tubes are more like collections, very large collections of axons that are sort of bundled together into these fascicles. And along the length of these fascicles, there will be both inside of these axons and outside of the axons, for the reasons that I showed you above. There'll be anisotropic diffusion along the length of the bundle rather than across. Yes?
What about dendritic connections?
So the question was, what about diffusion around dendrites? This is mostly, so dendrites are mostly present in the gray matter, where the cells are themselves. And in the gray matter, at the resolution that we're currently usually measuring, we don't really see a lot of anisotropic diffusion. So it's not really a good method for measuring very small parts of neurons in the gray matter. Rather, it's pretty good for measuring anisotropic diffusion along the length of white matter connection, in the white matter itself, and not in the cortex.
But a very quick follow-up.
Yes.
Gap junctions, the electrical junctions, much faster junctions are going to be dendritic, so it's like you can't actually factor it over there.
Yes, that is correct. So yeah, we cannot measure so well connections in sort of dense places where there are a lot of cell bodies. Here we're measuring these kind of big fascicles and the properties of these big fascicles. Yes. Okay, let me just tell you briefly how diffusion MRI works. So this is the measurement itself. Here now you'll see the scale is about 1 centimeter. This is a slice through the brain. This is the front of the brain. This is the back of the brain. And what I'm showing you here are different images that are acquired as a magnetic field gradient is applied across the sample. And it's applied in different orientations. So each image here in this movie corresponds to an image that was acquired when the gradient was applied in a different orientation. And what you can see is that the color of the image changes. So as we change the gradient, we're probing the diffusion in many different directions. And for example, if you look right here, what this part is a part of the brain called the corpus callosum. It's a big bundle of fibers that connects the two sides of the brain. And what you can see is that as we move this, as we rotate this gradient around, this part of the brain becomes brighter and darker. And that indicates to us that there's more diffusion in some directions than in other directions. In other words, the diffusion in this location is anisotropic. And we can take and process these images to tell us what the anisotropy is. And this processing is what I'll tell you about next. So the next question, once we make these measurements and we have measurements of the diffusion, the signal as the gradients are applied in many different directions. For example, here we have the measurements in one single volume of about 2 millimeters by 2 millimeters by 2 millimeters, sort of a little cube that we call a voxel. In that cube, we've made the measurement applying these diffusion gradients in 150 different directions. So these are 150 different dots indicating the distance. We've calculated the distance of diffusion in each one of these directions in this one volume. And you can see that this is anisotropic. There's more diffusion along this length than along this kind of boundary. And one way to model this is to try to estimate the parameters of a three-dimensional Gaussian distribution of diffusion, right? So we can draw that out as this kind of ellipsoid, right? So we take this and we fit a model with very few parameters that is sort of an estimate of the surface of this diffusion. It looks like a little bit like an ellipsoid. It is an ellipsoid. It's elongated along the axis of the most diffusion in this particular voxel. It tells us which direction there is most diffusion along, and might indicate to us where in what direction in that particular voxel there might be a bundle of these axons, one of these big fascicles. So that's how we model diffusion, and this model is quite useful for several reasons. The first reason is that we can actually estimate physical quantities of the tissue itself in that location. So what we do here is we take all these parameters and we estimate what is the mean diffusivity in all directions. And this is, again, a slice, sort of a horizontal slice through the brain. And you can see that there are these holes in here. These are called ventricles. They're water-filled spaces inside our brain. And in these ventricles, there's more diffusivity than, say, in the white matter here. So that tells us a little bit about the physical properties of the tissue in different parts of the white matter. Another kind of quantity that we can estimate is how much anisotropy there is. Anisotropy tells us, so this is called fractional anisotropy. It's a quantity that varies between 0 and 1. When it's 0, that means that there's pretty much the same amount of diffusion in all directions. When it's 1, it means that there's really a lot of diffusion in one direction, very little diffusion in other directions. And as you can see in big sort of bundles where there are these big connections along the white matter, the fractional anisotropy is very high. And then the third thing that I indicated before also is the principal diffusion direction. What direction is this ellipsoid the longest in? And this principal diffusion direction is mapped here as an RGB map. So we've transformed the x, y, z coordinates of the vector in which this ellipsoid is pointing into R, G, and B coordinates. And so red is places where the diffusion is elongated along the left-right direction. The green is along the front to back of the brain, and blue is up-down. So here, for example, there are bundles that go up and down to the cortex, and leading maybe, this is what we call the corticospinal tract. It leads, these are big bundles of connections between the cortex and the spine that transmit information about what muscles to move. So this is the primary motor output of the primary voluntary motor output of the brain. So that's this blue thing here. It might be the corticospinal tract. And then we can take this information that we extract from this principal diffusion direction, and we can do what we call fiber tracking. This is our way of identifying neural connections in living human brains. For example, let's zoom in. So here we have, again, a slice through the brain. This is one of these FA, this fractional anisotropy map. I zoom in on some part, and now I'm going to plot the tensor together with its principal diffusion direction mapped to this RGB map on top of this FA map. So you can see here, for example, there's high FA, and there are things, the tensors are pointing right to left. And that's because there's more diffusion along the right to left than along the front to back. And this is, again, this is where we know there is the corpus callosum, which is a big bundle of fibers that connects the two hemispheres. In another place, there might be more diffusion front to back and less diffusion on the right to left. And what we can do is we can propagate a streamline through the vectors defined by these tensors and estimate maybe a connection in this manner. And doing that over many different times, we get something like this. So here is, again, the corpus callosum. And notice this tells us not only that maybe there is a connection, a homologous connection between this part of the brain and another part of the brain here on the other side, but also what is the trajectory of these connections through the white matter, what their shape is. We might be able to probe maybe the tissue properties in different locations along this connection. It gives us a lot of information about the white matter and these connections. So it's a very interesting and valuable thing to do. That's all great. So what is the problem? Well, the problem starts with the fact that there are many different algorithms to do that. I showed you one model, but there are many ways to model the white matter, the directions of fibers within one particular voxel, and also many different algorithms to do the structography process. And these algorithms can produce very different results from each other. For example, we're looking here at the corticospinal tract that I just told you about, and another fasciculus called the arcuate fasciculus, this blue thing here. So this goes up, down from the cortex to the spine. The arcuate connects two different parts of the brain itself. And if you look at the results of algorithm 1, you see these smooth-looking bundles that don't intersect at all. Whereas if you take another algorithm, you might see very sort of tortuous trajectories that intersect quite a bit. And the question is, okay, what is in the brain? Is it this thing or that thing? Not only that, these different algorithms tell us quite a different story about the connectivity. This, again, is this first algorithm. If we look at the endpoints of the arcuate fasciculus on the surface of cortex, you might see quite different endpoints in one algorithm rather than the other. So this seems like a pretty serious problem. And the question is, how can we tell? Well, it's true that one of the major challenges that we face in MRI is evaluating and validating the different algorithms that we use. And it's also true that different features of the white matter might arise in different measurements. And so we think it's important to perform what we will call in vivo validation. What does that mean? It simply means we want you to evaluate the models of the white matter with respect to the data that you actually measured. Some people propose that it might be a good idea to take, for example, make these measurements on some animal and then open this animal and sacrifice this animal and perform some kind of tracer experiment. But the problem with that is that tracers are also not without error, and you have this data is already, you already collected this data. It might be good to actually validate this data in vivo on the subject on which you performed the experiment. And we'll talk a little bit about, I'll talk a little bit about how we propose to do that. One of the ways that we use is cross-validation, and it's a rather simple idea. If you measure two measurements, you can do one simple thing, which is just ask how does that measurement repeat itself? What is the test-retest reliability? So this, say, is the data in one voxel across 150 different directions of diffusion gradients. And we can compare it to the same voxel, same location in the brain, measured again with the same 150 directions, and just ask how reliably does this measurement repeat itself on subsequent measurements. We can compare that to another estimate, which is we take this data, we fit a model to it, and then we take this model and we try to predict the subsequent data. This is called cross-validation. And we can compare test-retest reliability to how well the model does in cross-validation. And so from this, we can calculate what we call a relative root mean squared error. That would be the error in prediction from the model itself relative to the error in prediction from test-retest. And if the relative root mean squared error is smaller than 1, that means that the model that we fit is better than test-retest reliability. And we think of it as a pretty good model of that particular voxel. So let's see what that looks like. So this diffusion tensor. DTI is often used to refer to this diffusion tensor, or this ellipsoid model, this very simple ellipsoid model that I told you about before. And what I've mapped here is again, this is sort of a horizontal slice. We have the 3 slices here, but this is sort of a horizontal plane. And we're looking at pretty much the one part of the brain that is not fit very well by this diffusion tensor model. Overall, it fits very well. It fits, the relative root mean squared error is smaller than 1 in about 97% of the voxels in the white matter in a typical measurement. So it's pretty good. But there are some places in which the relative root mean squared error goes above 1. I would note in particular this location. And this is no mystery to us. We actually know that this is a location where there are several different fiber bundles that intersect. So in this location, this is where the corticospinal tract and this corpus callosum going across, they both sort of intersect there, and there's another bundle that goes along the superior and longitudinal fasciculus that goes front to back in this same location. And the tensor might be a good model for, say, an individual fiber bundle going through the voxel. It's not actually a good model for a combination of different fiber bundles. So we need a different kind of model. And there have been these models have come up in the literature several times. People have proposed these kind of things several times. I kind of mentioned here all the times that we found that people have talked about this. You can model crossing fibers, what we call crossing fibers, as a combination of these tensors. So it's simply a combination. And well, we find that this kind of multi-fiber model fits the data better than the tensor model. It fits the data better in most of the voxels in the brain. And in particular, it solves the problem that we had in this particular location where we know there are crossing fibers. Accounting for these crossing fibers helps us fit the data better. So that's pretty good. But there's a sort of a second step to that, and this is, I should say, a work that was led by my colleague Franco Pistilli, who is now at Indiana University but was a postdoc in the same lab that I am in, Brian Wendell's lab at Stanford. And the project that he led was to try to not only account for the data in single voxels one at a time, but to account for this entire trajectory of an entire big bundle, or maybe an entire connectome together, right? So much as the model, the single ellipsoid model is a model of the white matter or the signal in a single location, these bundles are also a model of the white matter. And though typically what we do is we take the diffusion signal and generate these tracks in the way that I showed you before. We estimate them, we propagate these streamlines. You can also do it the other way around. You can use a forward model to try to predict the diffusion signal and then cross-validate on this diffusion signal. And so here's how that would work. You take, say there's one fiber here, you just put in there the prediction from a tensor in one location, then you go along and add the prediction from another location, etc. You go along and do that for every fiber. Say another fiber, you add back the prediction from each one of these individual tensors along the length of this thing. And you set up a big matrix. For each one of the fibers, you just put in the signal that would have been predicted along the entire trajectory in every one of the voxels through which this fiber or streamline passes. And each column here will correspond to one of these streamlines. And you can see that there are some locations where both of these streamlines, say here in the intersection, both of these streamlines pass through this voxel, and there's some kind of prediction from both of these streamlines. But some streamlines pass only through some voxels, and they don't have anything to say about those voxels in which they don't pass. So that's how we set up this matrix. And it's pretty large because we get a lot of these streamlines. And we just try to then fit this matrix to the data that we actually collected. So we set this up as a big set of linear equations. And for that reason, we call this method linear fascicle evaluation, or LIFE. So here it is. This is the LIFE equation. So it boils down to this kind of general linear model that you know from many different other things probably. And you can just solve for these weights. We solve for weights larger than 0 because you can't have a negative streamline in there. So it's a non-negative. We solve this using some kind of a non-negative least squares procedure. And then we can use cross-validation, predicting some kind of Helmholtz set using simply the predicted. So we've estimated the betas, the weights here as beta hat. We multiply back to get a prediction. And we can calculate the root mean squared error relative to the signal. Tell us how well, once we fit these weights, can we predict from this connection, from this set of connections together, the signal in another measurement. But the problem, so it works, it's great. I'll show you soon some kind of results from this kind of procedure. I'd just like to throw out there that this has some challenges coming with it. In particular, you end up with a matrix that's pretty large. It has on the order of millions of columns and on the order of hundreds of millions of rows. It's pretty sparse, but still it can be pretty challenging to solve this. It can be pretty challenging to set this up in the first place, and it can be pretty challenging to solve these kind of problems. So that's an interesting challenge. But it has its benefits. For example, comparing these two models that we talked about before, you can see that the error in predicting using a tensor is larger than if you use this multi-fiber, even on the scale of large fiber connections. So it actually tells us which one of these models of the white matter we should prefer, and which model of connectivity with the cortex we should prefer among the two. An interesting kind of side story to this, in a project that was led by Jason Whitman, who is a graduate student in the lab and now is actually at UW, so I'm following him up to Seattle. And Kevin Weiner is also a researcher over at Stanford. They were interested in this part of the brain. Jason, I'll just show. Jason, in some work he did a few years ago, he kind of discovered this interesting bundle that goes up and down in the occipital cortex, in the back of the brain. And digging back in the literature, they noticed that people had actually found this bundle. This is sort of late 19th century anatomy. People had found this bundle, but it sort of disappeared later. And one of the reasons that it disappeared, which is an interesting sort of sociology of science situation, is that this guy Maynard had an entirely different theory about how the white matter should be organized. He had a theory that long projection connections between different parts of the brain should only go front to back. And so he literally covered up, in his textbook that came out the year that he died, he literally covered up the occipital cortex with this vertical occipital fasciculus in it, and it pretty much disappeared from the literature, only to appear very few times in the scientific literature after that, until Jason made this kind of found it in his data, and looking back at the literature realized that people in the 19th century had identified this bundle already. Okay, and so we collected more data on this thing, and this is work that Hiromasa Takemura, who's another postdoc in the lab, led to say, okay, so we have this estimate of, we have these bundles that we say are the vertical occipital fasciculus. How well would we do in fitting the data if we assume they're not there at all? Right? So we do a virtual lesion, we take away these bundles, we say how well can we fit the data if those weren't there? And we can compare, this is sort of an error distribution, we compare two error distributions. One when it's lesioned, when we've removed the vertical occipital fasciculus, and one when the vertical occipital fasciculus is there. And we find that there's a large difference in the error, and you have a smaller error if you assume that the vertical occipital fasciculus is there. And this is sort of an in vivo validation of this anatomical structure, of the existence of this anatomical structure within an individual person. So we can say this anatomical structure exists, and we think that it's here. Here's the error on this estimate. Okay, so it has benefits. Now I'll move on to an even darker side of this, and that is that once we do these kind of experiments, it's very hard often to reproduce these experiments. And we all know this kind of situation from different scientific fields where data is quite expensive, complicated to collect. You spend a lot of time on collecting these things and spend a lot of money collecting this. This analysis requires multiple computational steps, some of them rather complicated. The new algorithms that people write are sort of in-house code or computational black boxes that you kind of download from somewhere without really knowing what it does. And this is not unique to MRI, as I said, but it is a big problem. And so the question is, what do we do to make this kind of research more reproducible and sustainable? Meaning in sustainability, I mean that people can continue each other's work. It seems like once we understand that we can find, say, the vertical occipital fasciculus in an individual, somebody else might want to also find the vertical occipital fasciculus in their individual measurement and maybe say something about the properties of the white matter inside the vertical occipital fasciculus in a patient or in some different individuals. And reproducibility has a particular kind of specific meaning here, which is that we think stems from this nice quote from Buckheit and Dono, that if we write an article about a particular computational result, somebody else should be able to just download that result and run it and examine it and look at the entire code. It should be all the software environment, the code and the data that produced the result. And so in the articles that we wrote based on these evaluations, what we try to do is to, first of all, make the data available, showing it through the Stanford library, and then show the analysis code itself on GitHub. And in at least one of these papers, every figure and every conclusion is supported by an individual IPython notebook. It looks something like that. Here's, for example, test reliability in one particular voxel over 3 different kinds of measurements. I haven't really gone into details about that. So somebody can download or clone my GitHub repository, download the data, and install the dependencies, and then run this. This is very nice, but it's not particularly sustainable in the sense that it requires maintenance, right? Code rots, as we all know. Over time, code that ran a year ago, we don't know if it will run tomorrow. It requires some kind of a larger community effort to produce this kind of sustainability. And so I'm going to tell you about MyPy, which is an ecosystem for reproducible neuroimaging. So this ecosystem idea is that we have the Python language, and people have written very good tools to do general scientific computing based on the Python language. And building upon that, we've built a series of tools to do specifically neuroimaging research in Python, including libraries to do time series analysis, libraries to deal with file formats. And for this talk, it's particularly interesting to focus on this thing. DiPi diffusion imaging in Python is sort of a particular part of that galaxy, which I'll talk about a little bit more now. Some of the principles behind this kind of community effort is that it's free, it's open source. We try to make it as thoroughly tested as we can. It goes through fairly rigorous code review through GitHub, the pull requests, etc. And that makes for a fairly natural place to collaborate around different ideas in neuroimaging. So you can propose different ideas for how to do neuroimaging analysis in this kind of environment, because it has the feeling of the kind of peer review that you want scientific ideas to undergo. And then we try to make it properly explained so that others can pick it up and start using it. So I'll show a little bit what I mean by properly explained. How do I do that, though? Let me see. There. Let's see what happens if I do that. There we go. Okay, so properly explained means, among other things, first of all, that we try to teach people how to use this software. But in particular, we have this whole system that we took from the developers of PyMVPA, which is a library for multivoxel, multivariate pattern analysis in MRI. We borrowed from them this idea that you could write, where is my mouse? Here it is. You can write these little scripts that include code and explanation together. So here, for example, is this linear fascicle evaluation now implemented in DiPy. Yes?
Can you make it bigger?
Oh, bigger, yes. Assuming I can get my mouse there, yeah. Ooh, there it is. So this includes both the code and a little bit of, we explain what we're doing here. It includes a code example, full code example, including, we're trying to evaluate the error in some candidate connectome. Here are the weights, fiber weights that I told you about before. Here are, say, distributions of errors, and that part of the brain, how well are we doing before we did this fitting procedure, after we do this fitting procedure. It has the full example here, and so somebody can then go and download, actually, the full source code. So that once you've installed DiPy, you can take this file, download that as well, and just run the entire example, which I think is a way to explain to people how to use the code, and gives them the first step they need if they want to actually continue this research that we've done. Now, how do I get out of here? Okay. I'll close you. I wonder what happened to my presentation. Mirror displays. One still wonders. There you go. All right. Okay. Now, so how do we know whether we're doing well? Are people actually using our software? One way to do that is to check how many people downloaded the software, and that's not a great way to do that because people might download it and then never use it again. Different people might download it more than once. It's not a great way to assess that. One proxy that was actually proposed by Jake in this blog post about why Python is the last language you will ever have to learn is that you look at the number of cumulative contributors to the library as sort of an extreme lower bound on the number of users. What Jake actually noted in this particular blog post was that as people started, as these projects here, which are sort of the core projects of the scientific Python universe, started using GitHub here in around end of 2010, the number of developers actually shot up, and so I extended that now, and you can see that Python is still the last language you will ever have to learn. I'll also add, so I wrote a blog post about this myself, and one thing to also add to this is IPython, and you can see this IPython, this curve starts to shoot up before these other curves start to shoot up. So maybe it's not only GitHub. It's worth mentioning that IPython moved to GitHub earlier than these other projects, maybe dragging all these projects along into GitHub. You can speculate about all these things, and I have. This is sort of maybe data science about data science. Might be interesting to some people here. But anyway, we can do the same thing with our, the numbers here are smaller, but also you can see, about 2 years time lag past these other projects. Around 2013, there started to be a large influx of more and more new developers, and that gives us some kind of a lower bound on how many users there are. There may be about maybe 30 people. Some of them may have gone on to do other things since, but it looks pretty good. It goes up. That's me here in 2011. That little bump there. I think Stefan is in here somewhere also. Yeah. Okay. And then finally, I'll talk about one more thing about sustaining reproducible research is also how do we get this into the system, into the way we do science on a day-to-day basis. I'll tell you a little bit about a project that we have at Stanford called the Project on Scientific Transparency. That is that reproducibility requires transparency. Those words are actually somewhat interchangeable. And in this project, there are sort of two aspects of reproducibility that are being covered. The first is about data sharing. And people at Stanford, I can't take a lot of credit for this, I can't take any credit, really. Gunnar Schaefer, Bob Doherty, and others at the Center for Neurobiological Imaging at Stanford have developed a system called NIMS, that stands for Neurobiological Image Management System. And the idea is pretty simple. In contrast to other databases where you do some experiment, you collect some data, you come to some results, and then at the end of the life cycle of the experiment, you go to some website and you upload your data. The idea here is that this system takes your data as it comes off the instrument, and this is your first point of entry at the very beginning of the process to get your data. It's not somewhere that scientists put their data, it's somewhere that scientists actually get their data at the beginning of the process. And it facilitates also the archiving and then the sharing, ultimately, of the data. It has a fairly flexible system of permissions on different pieces of the data, so different people can share with collaborators, specific collaborators, or just with the entire world, particular pieces of the data that they've used to do their science. The other aspect that we're thinking about is how can we do this also for analysis, and that becomes very complex and interesting and something we're thinking about a lot now, and I hope to be thinking about also in the future. One, we think, fundamental piece of the puzzle is, of course, the Jupyter project and everything around that. Another might be something that we're experimenting with our Docker containers and how we encapsulate pieces of our analysis into something that can very easily be downloaded, used in multiple different configurations. Maybe if your data is very, very large, you want to bring your analysis to your data rather than the other way around, wherever that data may be. This has a lot of potential to facilitate these kind of patterns. Okay, so that was what I had to say. I'll just summarize. I'll try to, again, bring this back to all of you, whether you're doing neuroscience kinds of experiments or thinking about other kinds of science. I hope that I showed you sort of a process where we go from the measurement of some kind of very specific physical quantity in the brain. We're trying to estimate a model about something that we're interested in, whether it be brains or other things, and we use statistics heavily to know that we've got the right model of that thing. But we also want others to be able to take what we've done and continue the work afterwards. We don't want it to just be a PDF on somebody's hard drive. It needs to be something that can be sustained. Finally, I'd like to thank my collaborators on all this work. Brian Wendell is the head of the Vista Lab where I am right now, a postdoc. I mentioned Franco, I mentioned Jason as well, who did some of this work. These people also collaborated on some of the work there. I have a very cool and interesting collaboration with Charles Zheng and Yuval Benjamini over in the stats department on some of these things. These are folks with whom I've been working on NiPi-related stuff. I should mention Fernando got me into this a few years back when I was a graduate student here and roped me in. And Eleftherios started DiPi, and these folks have helped work on that. Maybe if you do diffusion imaging, you would also like to help us, help yourself. And then this is the team around the project on scientific transparency. And also we have generous funding from all kinds of places. And I'd be happy to discuss this now if anyone wants. Thanks.
We have a full half hour for discussion and questions, so we welcome you to ask anything. And we are recording this, so Ariel will repeat the questions you're saying since we don't have enough in the microphone. Any questions?
Yes.
So one of the things that you said about the standard way of sharing data, which is to just upload it somewhere and maybe in a year it doesn't work. So you said that you can have this website and you put explanations and data there, but in 2 years when the software updates, do you update the code, or what is the solution to software?
So there are two different parts to this. One is the software part, and that is that what I've learned from my own sort of experience in working on this project, and just in the last few years, is that if you work by yourself on a software project that should do your entire analysis, it should do everything from the raw data to the figure, it's kind of hopeless, actually, because you won't be able to maintain that. So you need a community of people to do, you need to collaborate with a community of people so that the code doesn't rot. And we work a lot to make that code not rot. Matthew, sitting back there, is very active in really pushing this idea that we need to make sure that that this code works not only on my machine today, but also on some Windows machine that I've never even seen. So we have build bots that just go and check every commit into this repository. Does it run on all these different platforms? And that way we have a little bit more confidence that somebody else that was interested in doing this analysis or in examining my results doesn't have the experience of downloading the code, trying to install it, having it be broken, and then just moving on. Yes. Yes, please.
I direct science and technology for an Irish political party. I'm giving a talk actually at 3 o'clock, but the major issue I wanted to bring up, we're basically working on scrapping public funding of science, the NSF-style program. And your talk, I just wanted to address something in your talk as a result of that. So along with your project, your scientific transparency project, there's actually a project called the Berkeley Initiative for Transparency in the Social Sciences.
Okay.
This actually has some Stanford people on board.
Mm-hmm.
And I think this is exactly the problem. We've now got 2 different standards already of openness. So the Stanford people on board are people like John Ioannidis, who published that notorious paper, Why Most Published Scientific Papers Are Wrong, in 2005. And it is actually a serious paper with now, the question would be then, would your group be open to a dialogue with his group? This is particularly the case as MRI has been notorious ever since Ed Bull's paper, the voodoo correlations paper.
Okay, I'll try to repeat that.
Yeah.
The question was, I'll try to, you know what, I'll just pick out all that. I'll pick the thing I'm interested in. There are all these different initiatives of people trying to do reproducible, make science more reproducible. Why are we so split up in different groups, etc.?
Yeah.
Part of it is sociological, right? I mean, we're doing our own particular thing. The MINS system that I presented was developed originally to itch our own particular, to scratch our own particular itch. We had, or rather, people at Stanford, this was even before I came, had an instrument arriving, producing all this data, and they didn't want to follow the usual trajectory, which is you put this thing, and then all of a sudden you get this deluge of data, and you start figuring out where to put it. Instead, they wanted to prevent that by putting in place a system that will, even years later, be something that can be useful, right? I don't know if that addressed all of what you said, but certainly we know of Ioannidis, and there is obviously dialogue there. It's not we're not ignoring each other. So there is definitely we've definitely gone to there's this project they're calling Metrics over at Stanford. We've talked to these people. It's not like we're not living in different universes.
Okay, very quick follow-up. The MRI stuff, have you followed up the Ed Bull statistics? He did a meta-analysis of the fMRI results to date in 2009.
Yes, so you're asking about a paper that was published a few years ago where people did what might be called double dipping into their data. They detected some kind of a correlation, and they went to a particular spot, and they plotted that correlation and said, look how striking this correlation is. And yes, of course we have. The cross-validation approach that I presented here deals with that kind of issue completely. So doing cross-validation, I think the field, the MRI field, has definitely learned the value of this kind of approach where you separate your noise from your model. You try to separate your noise from your model by doing cross-validation. Yeah. Yes?
So if the human subject traveled from research group to research group that does this, and each time the subject is scanned, and then each group said, here's what we consider to be the raw data off the scanner.
Mhm.
How does that essentially reproduce, or do you do anything where you see very large differences between?
Yeah, so the question, if I understand it correctly, is how really quantitative is this measurement that you're making, right? If I measured an individual, if I took me and I measured me in Berkeley, and then I took me to Stanford, I measured there, and I traveled to Japan, I did this measurement again, how similar would the measurements come out in terms of, for example, if I had an estimate of diffusivity somewhere in the brain? And the answer is, no, this measure of diffusivity that I was talking about is not quantitative in that sense. It depends on calibration of the scanner and other issues. Our estimates of trajectories, some of them will be reproducible and some of them won't be reproducible. Also, that's kind of measurement noise. There are methods to measure MRI that are quantitative. There was briefly on the screen, there was a picture of my colleague Aviv Metzer, who developed a method to calibrate and quantify the density of tissue in different parts of the brain. That measurement repeats very nicely up to some level of noise, repeats very nicely when you take a different individual from one location to another. So it depends a little bit on what measurement you're doing. Diffusion is problematic in that sense.
It sounds like it's not ready for the clinic in that sense.
It's not what?
It's not quite ready for the clinic, right? If the doctor
Oh.
can't really do.
Yeah, it's actually, so the question was, it's not ready for the clinic then, and the answer is, well, it's being used in the clinic. For some uses, it's very useful, right? If you suspect that somebody has an infarct somewhere in their brain, a stroke, doing a diffusion scan is a pretty good way to assess that by looking at the diffusion scan, seeing if there's a big spot of higher diffusion somewhere. You don't really need to know if it's 3 or 3.2 to be able to do that, right? So depends on what question you're asking, always. Yes?
So you show this project where you store all the data from the MRI, and from that, people will get results. Are the results also stored in the database that you can query? And if at some point in the future the algorithm is improved and then those results are more detailed or changed, how would you deal with that and propagate that to the other analysis that would use this previous result for some other discovery?
Yeah, the question is, if you have a database with the raw data and you do some analysis on this raw data, now do you put back in the processed data into this database as well? And further, if that processing changes over the years, how do you redo the analysis to produce these other results again and again? And those are both really hard problems. The first is a hard problem because, well, as we've experienced in doing this kind of analysis, so the initial data, that one kind of interesting thing is that the data that we collect is not actually very large. The data sizes are not huge, but you start processing it. For example, you take the raw diffusion data measured at 2 millimeters on a side, cubes, and it's for the entire brain. Okay, you have some size of the data. And now you start doing this tracking process on it, producing 1 million different streamlines. All of a sudden, you have a huge file on your hand. So do I allow, if I have this database meant for showing results and data, do I allow any size of file to go in? And I don't actually know what the answer should be to that question. And the second question is also one that is very hard, right? What happens when you change your processing? Do you update the analysis? So here's the worst case scenarios. I did some experiment today. I fit some model to this data. 2 years from now, I discovered that I could do that better. Not that I could do it faster, but that I could actually be more accurate. Do I throw away the paper that I did before and update? I would say probably not, right? That's science. You keep getting it more and more accurate. So it depends exactly what changed, right? If there was an error, then if the code was just wrong and produced some result that wasn't there, we want to know that and we want to update things. But I don't think there's one answer to that question also. It depends a little bit Does that make sense?
Yeah, I was wondering if you were actually working on that or
No. Yes.
Sort of related to that, what about analyses that involve stochastic processes, right? So like the fiber tracking is often done probabilistically, and then you don't necessarily have an analysis that you can replicate exactly, right? So do you want to store like the outcome of that kind of analysis, or are you more likely to want to do that?
The question is, what about analyses that include some stochastic element, some probabilistic, so the tracking, for example, can be done probabilistically, whether or not to store that. So well, you can store the random seed, and then it would be reproducible. That would be one way to deal with that, but doesn't quite answer the question. I think, yes, I think you would want to store the results of probabilistic. So you did something. You did some analysis. It required some stochastic elements in it. You came to some results. It would be actually kind of great, given this stochasticity, if somebody else wanted to go and rerun it with their own random seed and see that the result still holds up. If it depends on the choice of the random seed, if the result depends on the choice of a random seed, the exact sort of the conclusion that you came to, rather than the exact result, up to some digits, is going to depend on the random seed. But if the conclusion that you came to depends on choice of random seed, that's something we want to know. And so I'd say, yes, store that thing, and then have others do it again, reproduce it, and try to see what that stochasticity did. If that makes sense, I don't know. Yeah. Yes.
I have a question about this NIMS repository where the data are coming in from the machine. I like that idea of putting the kind of repository responsibility a lot earlier in the data lifecycle. Are you finding people coming in from other fields asking if they can kind of copy that model?
So the question is about the database model where the data goes in straight from the instrument into the database, and sort of scientists get their data from that, they don't put their data there. And the question is whether scientists from other fields have shown interest in this. And that's a good question. I'll defer to others in the team to answer that. I don't actually know whether there has been, but obviously it has application in many different fields. I will say that scientists that use this system on a day-to-day basis, I've been around them now for a while, don't think of this as a system for sharing data. They just think of it as the place they go to get their data when they want to do their analysis. And that has huge benefits, right? Because then once you go to them and you mandate sharing, for example, somehow, and you tell them, oh yeah, no, you're already set up to do that. You don't need to do anything. You just need to click on this checkbox that will just make this public. I think that has a huge benefit for people. Yeah. Yes, please.
In your experience, how prevalent is the attitude that, I've worked for many hours on my code and I don't necessarily want to share it because, copyright and patents aren't used the same way in science as in many other fields, and someone can just kind of take it and not necessarily give credit. So is that attitude really common? Do you think that's part of what's blocking a lot of efforts for reproducibility, and how would you combat that attitude?
Yeah, the question is, and there might be others here who could also answer that, I'm sure. The question is, how prevalent is the attitude? I've worked on this thing. It's pretty valuable, I think, and I don't want to share it. I want to sell it. I want to, I don't know, I want to use it again before I make it available. Right? If I've collected this extremely complicated data, very large data set involving many subjects, I'll go to data with your permission, but it's true for code as well. Should I make that? I've just published a paper on it. Should I make the code of the data available? I want to write like 5 more papers on this data set before I make it available. That attitude is prevalent. I've seen it around, and I think, to some degree, I actually respect people's right, people's copyright to their code, people's right to their data. I respect that to some degree. It's not clear exactly where you put that limit. If people make certain scientific claims about the brain, for example, using paying for the experiments with taxpayer funds, I'm not so sure, where the line exactly crosses. But I would like to respect. I would like for people to not be demotivated to do cool things just because of this feeling that they have to share. That said, I think there's additional value that can be elicited from the act of sharing. The typical thing is, for me, as somebody who makes a lot of errors, is that if I share my code, others will come along and make it better for me. So I like that. Saves me a lot of time and pain. Yes, Matthew back there has.
It seems to me that once you've gone into an atmosphere where you've seen some collaboration of people sort of read your stuff, they check it, they say whether it's good or not. It seems, at least to me, it seems so much better a way to work, so much closer to what I thought science was, that it's very difficult to imagine going back. Do you have experience? Have you seen people sort of do open source? And I think I don't know if actually sort of keeping the code to myself is a better idea.
The question was, have I seen people do open source and then say, you know what, this project is pretty cool. I think I'm not going to tell anyone about it until, yeah, sure, I've seen that. Yeah. I'll say more about that. I've learned a lot from doing open source software development myself. So I'm sharing code, and I've just learned so much from it. I think I've gotten so much value out of it that I don't entirely understand. When people come to me and say this kind of thing, I say, well, I respect your attitude, but it's you're kind of shooting yourself in the foot. It's probably not as valuable as you think it is if you're going to do that. Yeah.
So let's say this is one of the things that is a little bit in the way of reproducible research, and then maybe other things. What's your take on what are the main things that you think are in the way, and what do you think we should be doing to address? What's the first thing you could say to the group? Okay, guys, this is one idea that we should try to implement to make reproducible research better. What would be your first case?
Thanks, that reminded me. So the question was, what are the obstacles that we're facing to make research more reproducible? Well, what should we do? And that reminded me that I wanted to mention this really interesting article that was in PNAS this week, actually. This was Roger Peng and JT Leek writing about reproducibility and replicability. Those are two different things. Reproducible means I'll give you my data, I'll give you my code, you run it on your computer, you got the same figure that I got. Replicability means you go do the same experiment on somebody else. Right? You take some new subject that we haven't measured the vertical occipital fasciculus for, and you do the experiment, you find the vertical occipital fasciculus is right there in that person too. That's replicability. And that's actually what we aim for, is replicability of our results. Reproducibility, a reproducible result can be just wrong, right? If you have a bug in your code, it's reproducible. That bug will come up every time. And they point to something that I think maybe answers your question, which is education. That you want to prevent. You want the scientists to be sufficiently sophisticated in looking at the data so that they can evaluate how well they're doing in terms of reproducibility. And I think that's really important. I think we've had sort of in the lab, after reading this article, I sent it around to people and we had a little bit of a discussion. And we have disagreements about this among us as a group. We all think reproducibility is really important, and we're not really sure what to do on replicability, obviously, and we're not really sure what is the right thing, right? If the incentive structure is all wrong, right? Should we make the principal investigators, the people that are sort of the powerful people in the field, feel more strongly about this because they don't really need to feel strongly about that right now. Their livelihood does not really depend on caring about these things right now. Or should we just go to, in my feelings, we should go to the young ones and convince them that they don't want to be in a field that doesn't, where you can do an experiment, you can publish it, and then somebody else comes and it just doesn't hold up, right? It's, so that's my feeling. And it's particularly, I think it's particularly strange in, I'm in a psychology department, right? And a lot of researchers in neuroscience come from a background in psychology, and psychologists, myself included, rarely get trained in computational research. Many scientists, I shouldn't just say about psychology, don't get trained in computational research, in computational practices. I think things like software carpentry, obviously, play an important role in making this, making everything, raising the level a little bit. I don't think there are like structural impediments, right? There's no like, oh, this kind of research could never be reproducible. It's complicated, right? There are all these technical things that need to be figured out. Like if you want to do like the questions before about if you do some pipeline and it's really complex and you put together several steps, how do you make sure that you always have the same steps in place? But that's just the technical issue. I don't think it's like a structural, fundamental impediment.
Thank you very much.
Thanks.